Welcome everyone to our second talk of today's session. We hope you're all enjoying Horizon so far. There is some small announcement we have to do before we get started with our speaker. Please go ahead and vote for your favorite poster by going to the handout section and then just click on the poster voting file. And then also we have our closing ceremony at 6:40 p.m. So I would like to see you as many of you there. And now, so it is my great pleasure to be welcoming today Rubén Fernández-Busnadiego on Horizons 2020. Rubén studied physics at the Complutense University of Madrid back in Spain, and then shortly after he did an internship with Marcellus Ubink in Leiden University in the Netherlands, which sparked his interest in structural biology. With this, he then moved to Germany, where he carried out his PhD in the department of Wolfgang Baumeister at the Max Planck Institute of Biochemistry in Munich. And there he used cryo-electron tomography, for short cryo-ET, to study the structure of the presynaptic terminal, investigating the roles of the presynaptic cytomatrix in neurotransmitter release. Seeking to deepen his knowledge in neural and cell biology, he joined the lab of Pietro De Chemili at Yale University at the USA, where he investigated various aspects of membrane biology. He then returned to Munich to establish his own group at the MPI of Biochemistry with the aim to employ cryo-ET to unravel the mechanisms linking protein aggregation and cell death in neurodegenerative diseases. Since 2019, he's now a professor at the University Medical Center Göttingen, where his group continues to harness the latest breakthroughs in cryo-ET technology to decipher the inner mysteries of the cell. And related to this, he has open positions for both PhD students and postdocs. So if you would like to join his lab, please write to him. I guess he will tell you more about this at the end of his talk. And so with this, I give a warm welcome to Rubén to our virtual stage, and I'm just looking forward to your talk.
Great. Thanks a lot, Julio, for the introduction, and thanks a lot for the invitation. It's a great pleasure to join you guys today. Of course, it would have been much better to be able to do this in person, but that's how things are, right? So let's get started. I want to tell you a little bit about the things we have been doing in the last couple of years using cryo-electron tomography to understand the molecular basis of neurodegenerative diseases. So imagine you could zoom into the cell with such magnification, with such degree of detail, that you could actually see how cellular complexes are carrying out their functions. And now imagine you could do this actually in a disease-relevant situation. You could then directly see what's wrong with these proteins, with these molecules, and then this could directly suggest or open up opportunities for new therapies that would directly address the molecular causes of disease. So this is what my group has been trying to do using cryo-electron tomography, as I said, for the study of protein aggregates in neurodegenerative diseases. So I'm pretty sure you guys all know, for more than 100 years now, we know that the brains of patients of different neurodegenerative diseases contain these large aggregates of misfolded proteins. But after 100 years or more, we still do not understand the role that these aggregates play in the disease. So we don't really know if they are really the cause, or are they more a side product of the really toxic pathways. We still don't know.
Rubén, sorry for interrupting, but I think our attendees cannot see your screen for some reason. And I think, are you switching slides now? Because we cannot.
Yes.
Okay, then this actually doesn't work now. Maybe you can try to reshare your screen. Sorry for the.
Do you see it now in presenter view?
It's loading. Yes, we see it. We see it.
Okay, so then I continue. So what we want to figure out is basically what is the structure of these aggregates, but we want to understand their structure within their native cellular environment. And the main questions we want to address are two. First, what is the in situ structure of these aggregates? Now, we have structures of many aggregates that have been either made recombinantly or purified from the brains of patients. But these proteins, these aggregates, are extremely polymorphic, so their structure is highly dependent on the environment in which they form. So that's why we want to look at these structures within their native cellular environment. But perhaps most importantly, what we really want to know is how do these aggregates interact with the rest of the cell? Because we postulate that if we understand these interactions, we may start to gain clues about the toxicity mechanisms, about how do these aggregates contribute to disease, if at all. So I'm going to briefly introduce you the technical workflow that we use to address these questions. First, we need to vitrify our cells. So all sorts of cryo-EM rely on vitrification. This is a process of very rapid freezing that turns liquid water into an amorphous solid. And it's so fast that it avoids the formation of crystals. The water molecules do not have time to rearrange in a crystal lattice and stay with a molecular organization similar to the liquid state. So to do this, we grow cells, for example, primary neurons, directly on EM grids that are covered by a carbon support. And then we plunge freeze them into a cryogen that's cooled by liquid ethane, by liquid nitrogen. So this is what you see here.
Rubén?
Yes.
Sorry again for interrupting, but we cannot see the plunge freezing. So I guess maybe you are in the full screen mode, but we cannot see this. What about trying to share actually your whole screen instead of a window application? This might work better.
Okay, so that's what I'm doing now. Do you see it now? See the movie?
Yes, we see it. Yeah,
okay.
Yeah, it moved. Yeah. Thanks a lot. Sorry for interrupting again.
No worries. So then the sample goes very quickly into this cryogen, and thereby it's vitrified. Once we have the cells vitrified on an EM grid, what you see there, we can do cryolive microscopy. So once the sample is frozen, we must keep it at liquid nitrogen temperature all the time, throughout all transfers and throughout all imaging. So we have these microscopes with a cryostage that keep the sample at liquid nitrogen temperature during light microscopy imaging. And there, we can zoom into the grid and look for structures of interest. So for example, here we see one cell with this fluorescent dot, and this is a fluorescently tagged aggregate that we then want to study in more detail. So in principle, we could already look at the cells in this state, but they are too thick. Because in the end, we will do transmission electron microscopy, so electrons have to go through the sample in order to reach the camera. And if the sample is too thick, the image is just going to be too noisy. So we need to make the cells thinner than they are. And the best way to do this with vitrified specimens is a technology called cryofocused ion beam milling. I hope you can see the movie. What you see there is a cryofib instrument. It has actually two beams, an ion beam and an electron beam. Now the sample is being loaded into the microscope. There is this shuttle with two EM grids. We are seeing now a technical step that's not very important for today. The important thing is now we're going to use the scanning electron beam to locate the same cell we identified by light microscopy. Okay, and this is we're gonna zoom now into that cell, which is, as I said, growing on this holy carbon support of the EM grid. And this cell contains the aggregate that we want to study. We are now gonna use the ion beam to remove all the parts from the cell that are below and above this aggregate. everything that's on the way for our transmission electron microscopy imaging. So this is how it's done. You know, in reality, it's not such a drastic procedure. It's kind of a more careful, stepwise approach. But in the end, what you see there is what we get is this very thin slice of the cell that's suspended from the rest of the cell that has not been milled away. And if we did our correlation right, our structure of interest is contained in that, what we call a lamella. We then transfer the grid from this focused ion beam microscope to the transmission electron microscope. And that's where we do tomography, meaning that we take images of the sample from different angles by tilting the specimen stage. We record the so-called tilt series, which in reality has between 60 and 100 images, and then we computationally reconstruct those images to reproduce a three-dimensional volume that was contained in our cellular slice. So now you will see some examples of polyglutamine inclusions, which are related to Huntington's disease and neurodegenerative disease, which we studied in this case, for example, in a HeLa cell. You see now the tomogram, and in a moment we will see the three-dimensional rendering representation, where you can see all these Huntington fibrils in blue, and also all the membranes contained within the aggregate and its periphery. Now we're going to take a look at essentially the same thing, but in a primary neuron, and a little bit of a lower magnification, so we can see the complete perimeter of the aggregate. But it's essentially the same thing. You may be able to appreciate this kind of radial organization of the fibrils in blue, of the Huntington fibrils in blue. And we see that there are a lot of vesicles, these white structures within the aggregate, and the whole thing is surrounded by the red membranes, which are ER membranes. We can detect macromolecules, for example, ribosomes, which are shown in green. And we see that although there is definitely room for them to go into the aggregate, they don't. They stay away. What we found in this case was that when we would zoom in into places of close contact between the fibrils and cellular membranes, we saw structures like you can see on the left, in which the fibrils contacted the membranes and induced very high membrane curvature. And what we showed in this paper, but I will not go into today, is that this induction of very high curvature on cellular membranes is likely to lead to their disruption. So we think that these Huntington fibrils are actually breaking the membranes of cellular organelles, mainly the ER. And we think that this is then an important contribution to the toxicity that these aggregates cause in the neurons. So this was about this Huntington's disease. I will tell you now a bit more in detail about another study we did a couple of years ago, which is related to amyotrophic lateral sclerosis, or ALS, and also frontotemporal dementia. So a mutation in the C9orf72 gene is the most frequent cause of familial both ALS and frontotemporal dementia. And what happens with this mutation is that there is this GGGGCC sequence, which is in principle in an intron of the gene. But upon mutation, this sequence gets repeated an amazing number of times, up to thousands. And when this happens, although it's intronic, it's actually transcribed and translated. But translation, because there is no star codon, translation happens in every possible reading frame and in sense and antisense directions. And this leads to the translation of the so-called dipeptide repeat proteins, which you can see on the bottom left. These are the results of reading that sequence in every possible frame, so poly-GA, poly-GR, etc. We know that the aggregates that patients have in the brain have mainly poly-GA, and we know that poly-GA alone can cause toxicity in neurons. So we think this might be one of the main drivers of disease, these aggregates. And we wanted to take a look at how would they look inside the cells. So for that, we applied again our workflow. We grew primary neurons on an EM grid. We expressed our aggregating protein, in this case poly-GA, in these neurons, which is fluorescently labeled, and this allows us to do correlation microscopy. So in panel B, you see a vitrified neuron on an EM grid, and it's the SEM image is overlaid with a cryolight microscopy image. We see that it contains this green aggregate. In C, you see the same cell, but now from the side, from the perspective of the ion beam. And the yellow boxes mark all the material that we will remove with the ion beam. And this will result in the very thin slice that we see in panel D. In panel E is the same slice, but now transferred to the transmission electron microscope. We see there is some contamination, but the area that contains the fluorescence is still there. We can record data. And we can take tomograms such as this one. So here in these tomograms, we show them typically we go up and down optical slices. So we can see the network of the aggregate. And maybe you can appreciate that between these fibers, there are a lot of very regular structures. So the advantage of using these techniques is that we can recognize molecules directly from their structure inside the cell, because their structure is so well preserved that we can recognize it. And what you see in this movie now is the result of that process. So we see the tomogram again, we go up and down, and now we're gonna show the main molecular species that we found in this volume. So in red is the aggregate. It forms kind of planar ribbons. And in yellow, you can see ribosomes. You can see they are mainly at the periphery of the aggregate. The same is true for these purple spheres. These are the trig chaperoning. But what was really dramatic here, in this case, was the accumulation of the green particles within the aggregate. And it turned out that these green particles are 26S proteasomes. We measured that the concentration of proteasomes within the aggregate was around 30 times over control concentration. But at the same time, we saw no increase in the expression level of the proteasome in these neurons. So this means that a large fraction of the neuronal proteasomes are actually sucked into these aggregates, which act like a sponge. And we found this very interesting because, of course, proteasome dysfunction is implicated in many diseases, particularly many neurodegenerative diseases. And also because it's known from previous studies that this type of GA-rich peptides impair proteasomal degradation. So we wanted to zoom in into this phenomenon, and to explain you what we did, I want to tell you a little bit about the structure of the proteasome. This is coming from single particle cryo-EM studies from the group of Eri Sakata and others. The proteasome has two main components. One is the core particle that contains the proteolytic chambers, and the other one is the regulatory particle, which binds ubiquitinated substrates, unfolds them, and translocates them into the proteolytic chambers. And proteasomes can have either one regulatory particle bound or two, one on each end. And we know from the single particle studies of our colleagues that the conformation of this regulatory particle tells us about the functional state of the whole machine. So what we can do now with our cellular data is that we found this bunch of proteasomes inside the neuron coming to this aggregate, and we can then extract them computationally one by one and treat these single proteasomes as essentially single particles in single particle cryo-EM. So we can do averaging, we can do classification, and we can sort them out in groups according to their structural conformation. So this is what you see here. We found 4 main groups. As you can see, the resolution we can achieve, or we could achieve in this case, in this in situ study, it was between 11 and 15 angstroms. This is not as good as you can do with purified proteins, where you can go almost or all the way atomic. But it was definitely, but I mean, one has to remember that these particles were not purified, were imaged within the cell. And this resolution is definitely good enough to dock existing atomic models that we have from our single particle colleagues. So from the single particle work resulted in atomic models of the different functional states of the proteasome. And now we can see which of these atomic models maps best with the conformations we found inside neurons. So this comparison revealed that these 2 green classes corresponded to the so-called S1 state. This is a ground state in which the proteasome is essentially inactive. This other class corresponded to the S2 state, in which ubiquitinated substrates bind the proteasome but do not start translocation yet. And what was most interesting for us was this last group, which we mapped to the so-called S4 state. We found this very interesting because the group of Eri Sakata showed in vitro that this S4 state is a state of active translocation of substrates. So the substrates are being degraded by the proteasome in this conformation. But at the same time, from the in vitro work, we know that this is a very transient state. However, in our cellular data, we found that almost a quarter of the proteasomes, so 23%, were actually in this particular conformation, meaning that in this context of the aggregate, this conformation was much more abundant than it was supposed to be, suggesting that maybe the proteasome just gets stuck, it's stalled in this state. So we wanted to investigate this in more detail. And what we can do with this data is, so what I told you before is we image the proteasomes within the cell, we extract them and figure out their conformation. And now we know the functional state of each individual proteasome. And we can put them back into the cell and see if there is any spatial pattern of organization of these different functional states. So one of the things we did was to measure the distance between each individual proteasome and the closest bit of aggregate. So this is what you see in the graph. And there are 4 curves with dotted lines. And each of these dotted lines is one of the 4 states that I just showed you before, these 4 groups. I wanted you to look to the cyan line, which is this S4 state that we found most interesting. And as you can see, if we look at proteasomes that are within 15 nanometers to the proteasome, almost 40% of them are in this particular state. And just to give you a sense of scale, one full proteasome is 40 nanometers long. And the way we are measuring this, when we say one proteasome is within 15 nanometers to an aggregate, it means it's essentially touching, like the ones you see on the left. So these are proteasomes that are directly engaging the aggregate. Those guys, almost 40% of them are in this particular conformation. And now if we move away by only 30 nanometers, meaning less than the length of one single proteasome, the fraction of proteasomes in that state already goes down to half. So this strongly indicates that it is the direct interaction between the aggregate and the proteasome that stabilizes this particular conformation, which it's supposed to be very transient. So again, it reinforces the idea that the proteasomes are really stuck on this aggregate. So what we think is happening here is that they're trying to chew on the aggregate, but it just doesn't work. And they keep going, but the aggregate is not being degraded and the proteasomes don't let go. And finally, to test this hypothesis that the proteasomes are impaired, we can do functional assays that indeed show us that there is an accumulation of proteasomal substrates in the presence of aggregates. So indeed, proteasomes are impaired by their interaction with the aggregates. So just a little intermediate summary of what I told you so far. I showed you these 2 types of aggregates. The first one, or what I just told you about, are these poly-GA aggregates related to C9orf72 mutations that cause ALS and frontotemporal dementia. And those guys, what they do is basically sequester these crazy amounts of proteasomes and get them stuck. What I told you before, a little bit, very briefly, was about polyglutamine inclusions that are related to Huntington's disease. And we see that those guys do not care about proteasomes, but actually they are very sticky to membranes, and we think they even break membranes. So really, each of these aggregates seems to be a completely different beast. So now I want to move to most recent work on Parkinson's, or related to Parkinson's disease and other diseases characterized by the aggregation of alpha-synuclein. So there was a very recent study in which they did electron microscopy, but let's say old style, so room temperature microscopy of plastic embedded samples, but this is of patient tissue. And what they found there is that aggregates of alpha-synuclein, so-called Lewy bodies, look like this. They are just like a bunch of organelles coming together And in this study, they couldn't see many fibrils. There is one little fibril there marked by this pink arrow, but they couldn't see them well, and they didn't really know what they were, and so they didn't really care much about them. So actually they proposed a model saying that these aggregates are basically just a bunch of organelles, and that there is synuclein in between them, but probably just little bits that is acting as bridges between organelles, so that the synuclein is really clustering these membranes together. And this has caused quite a bit of impact in the field. And there are some people that propose that we should now move from a fibrillar model, in which we think synuclein is really making these fibrils, to this other membrane-rich model, in which synuclein is maybe just building these bridges between organelles, and this is what clusters them in the Lewy bodies. So we wanted to test these models, but also understand better how do these pathologies spreads during the brain, because as you may know, many of these diseases start in a particular location of the brain, and then they travel normally along the anatomical connections and spread to the whole organ. And the way this is supposed to happen is that proteins misfold in the cell and aggregate, and then these aggregates somehow get out of the cell and are taken up by neighboring cells. They are endocytosed. So we wanted to understand this process better as well. For that, we used a model of synuclein aggregation, in which we basically culture neurons and we feed them with synuclein aggregates that we produce in vitro, recombinantly, but also from patients. I'll show you in a moment. And when we do this, and as you can see on the right, if we throw these so-called preformed fibrils or PFFs to neurons, they develop these small aggregates that you may see here in the phosphoalpha-synuclein channel, these little dots here. If we simply overexpress synuclein, but we don't seed aggregation with exogenous aggregates, there is nothing. There is no aggregate formation. If we both overexpress synuclein and seed with preformed fibrils, then there is massive aggregation that you can see in the bottom images marked by phosphosynuclein. So phosphosynuclein is actually a marker of Lewy bodies in pathology, and our aggregates are positive for it, and many other markers that are actually found in the brain of patients, and we know they are also toxic, so neurons die when they develop these aggregates. So we wanted to know what's going on. We zoom in into these aggregates using this workflow, and this is what we find. So we find lots of organelles as found in patients, but the organelles are really intermixed with lots of fibrils. So this is a still image of the same thing. And the fibrils are marked by these red arrows. And as you can see in panel B, they seem to be decorated by these globular densities. And they definitely look very different from, for example, actin that you can see in C, or microtubules that you can see below. So are these fibrils actually synuclein? Well, we can say for sure, yes, they are, because these additional green densities that we found, these were in cells that were expressing GFP alpha-synuclein. If we look at cells that express endogenous synuclein, we see, as you can see on the right, we see in panel F that the fibril is essentially the same as in panel B, as with GFP, but it doesn't have any more these globular densities. So these globular densities are directly GFP. This tells us that we have enough resolution to directly see the GFPs on our fibrils, and conclusively shows that the fibrils that are in between the organelles are actually alpha-synuclein. This was done with recombinant fibrils, but we can do the same with fibrils derived from the brain of a patient, in this case, a patient of multiple systems atrophy, which is also characterized by the aggregation of synuclein, and everything looks the same. So how does the seeding work? How do these aggregates get into the cell? To address this question, we labeled with gold aggregates in vitro, and we fed these gold-labeled aggregates into the cells. And what we saw is shown here. So hopefully you see in panel B, these gold-labeled seeds. The gold particles are marked by orange arrows. And from these seeds, there are fibrils marked by black arrows that are growing, okay? So what we found is that these seeds only have a small number of gold particles, typically between 3 and 10. And then the fibril grows from there. So this is telling us that actually what's being taken up by the cells, what's the most seeding competent species are kind of bigger oligomers or smaller fibrils that can only host a small number of gold particles, but that can then trigger aggregation of large fibrils inside the cell. A big question was, do the fibrils interact with the membranes like we saw in the polyglutamine case? And this is important, particularly important here because synuclein is known to interact with membranes. It has an amphipathic helix on the N-terminus that's known to bind membranes. So do the fibrils also mess up with membranes in this case? When we looked at the interaction between fibrils and membranes, in many cases we saw that there was just no interaction. Sometimes they come close, but nothing happens. But sometimes it looked like something happens. Maybe they seem to be pulling a little bit. We see that membranes appear to be deformed when fibrils are closed. But we also see this deformation in the membrane sometimes where there is no fibrils at all. So, you know, how this was not straightforward. And, you know, how can we figure out, is there a real interaction or not? Well, the nice thing here is we can just measure it. We have all these computational tools. We can extract the different things we're interested in from our data and just measure it. So we have the fibrils, we have the membranes, and what you see there is a color coding of fibril to membrane distance. We can then measure this, and this is shown by the green line. And the idea here was, well, if these contacts between fibrils and membranes are really something substantial, if we computationally move around the fibrils, we will lose the contacts, okay? Because this is really a biologically driven phenomenon. If this close proximity between fibrils and membranes is random, when we do a random simulation, we will not lose it because it's still there. And this is exactly what we saw, that if we shift and rotate the position of the fibrils computationally, if we move them around a little bit, actually the short distances to membranes became more abundant than in the experimental data. So this tells us for sure that the interaction between fibrils and membranes is not substantial. It's something that happens probably just due to cellular crowding. But there's also the question, okay, maybe it's not the fibrils, you know, but maybe this membrane clustering is happening by smaller alpha-synuclein organizations. Maybe it's even the monomers, like shown in this paper, that can actually bridge to membranes and bring them together. And these monomers are too small for us to see them. So could this be the case? What we did then was to simply measure the distance between membranes. Are membranes closer together in all these conditions in which we have synuclein aggregation than in control cells that have no synuclein aggregates at all? And the answer is no. So the membranes are just as close to each other in all conditions. So this means that synuclein in either form, fibrils, monomers, oligomers, anything, synuclein does not induce the clustering of cellular membranes. But then what's wrong, you know, because we know that these cells die when they develop these aggregates. So what's wrong? Why are all these all these membranes in these aggregates if they are not brought in there by synuclein? Well, what we have seen is that these membranes are not just any membranes. So if you look at the color code here, you'll see that these purple membranes are particularly abundant. So these are endolysosomal organelles, and this we see also a lot of autophagosomes or autophagic structures, which are marked in cyan. And I'm not sure if you see my pointer, but here on the left, you see an image of one of these autophagosomes, this C-shaped structure that's actually engulfing cellular material. So we see that there is a dramatic accumulation of these particular organelles, and we actually have functional data that tells us that both perhaps the autophagic and the endolysosomal function is impaired in these cells. So this is what we're basically investigating at the moment. The jury is still out. But I'm just going to stop here and summarize that what I told you before, that neuronal synuclein aggregates do not interact with cellular membranes directly, but they seem to mess up with the endolysosomal autophagic system. Altogether, what these studies really show us is that each of these aggregates in each of these diseases is it has a different architecture, and it interacts with the cell in a completely different manner. So if we are ever going to treat this aggregation in these diseases, we may need to do it in really a differentiated way for each specific syndrome. And finally, perhaps as an overarching conclusion, I hope I convinced you that we can use cryo-electron tomography to look at cells in an exceptional state of preservation and with molecular resolution. So I just want to thank the people who actually did the work. This was all done when we were still in Munich in the Max Planck of Biochemistry. Felix, during his PhD, did the study of the polyglutamine inclusions. Chang, during his postdoc, looked at the poly-GA aggregates. Now he's opening his own lab in Beijing. And the last data I show you is from Victoria on the synuclein aggregates. And finally, I mean, as I told you, there are still many open questions on many fronts, but particularly on the synuclein front. And now we've just been awarded with a big grant. So all this money is not just for us. It's a big network of consortium teams from all over the world, 21 teams from all over the world, that is really coming together to understand the molecular and genetic causes of Parkinson's disease. We are lucky to be one of these teams, and we are excited to welcome new people, both PhD students and postdocs, to look into these topics and into many other topics that we're interested in. Thank you very much.
Thanks for the very exciting talk, Rubén. We got quite a few questions. So the first one comes from Claudia. She will actually, she's the next awarded student talk, by the way. So she asked, can one do tomography directly on tissue rather than culturing neurons on grids, for example, from patient brain slices?
Yeah, so that's a big frontier. The answer is now not, for two main reasons. One is when the sample gets thicker, it's not possible to vitrify them so easily anymore by plunge freezing. We need to do high pressure freezing, and coupling high pressure frozen samples to cryofib milling is very complicated. So it's possible, but it's very complicated. The other reason is that if we take patient from tissue from a patient, we are actually breaking the cells. If, for example, we would take a biopsy, we're actually breaking the cells with our needle. So, and then we are looking, if we would look at that by tomography, by cryotomography, we are looking at a situation in which the sample has been distorted already. The cells are probably dying already just by extracting the sample. So I believe, this is what we need to do in the future, but it's not straightforward.
Mm-hmm. Great. So now CD is asking, have you done any biochemistry experiments to validate this fibril to membrane interaction? Do you also do the tomography for mutants lacking the membrane binding domain?
Sorry, what was the first question?
The first question is if you have you done any biochemistry experiments to validate this fibril to membrane interaction?
No, we haven't done it, but there is a lot of work already from other groups that have shown that synuclein can bind to membranes in vitro, but we haven't actually tested deleting the membrane binding domain, because I mean, in this case, we do not see any membrane binding. So I think, if we delete the membrane binding domain, we will still see no binding probably. So yeah.
Right. And the second question was, do you also do tomography for mutants lacking the membrane binding domain?
Yeah. So this is what I say, that because we don't see an interaction of synuclein fibrils with membranes, I don't think that deleting the membrane binding domain would be very insightful. But this is something that we could do, for example, and we haven't done yet with Huntingtin, which does interact with membranes. There are 17 amino acids on the very end terminus of Huntingtin that are thought to interact with membranes, and it would be very interesting to see if we get rid of those, do the fibrils stop messing up with membranes around Huntingtin inclusions. Yeah.
Mm-hmm. And the next question is also from CD. She asked, is it just the stalling of the proteasomes, or do you think the aggregates stabilize the S4?
So yeah, we think that by stabilizing the S4, the proteasomes are stalled. So these GA-rich substrates are actually known from some viruses, and some viruses like the Epstein-Barr virus uses a protein that's rich in similar sequences to escape from proteasomal degradation in the cell and to stay as a latent infection. So the idea is that these peptides are kind of slippery, these GA-GA repeats, and that the ATPase motor of the proteasome that has to bring the proteins down the chamber for proteolytic degradation actually doesn't manage to grab it. So it's kind of pulling from it and pulling, but it doesn't manage to translocate it, and therefore there is no degradation.
Okay, thank you. The next question is from me. So I'm wondering whether you have in your pipeline to treat these cells, these neurons in this case, with drugs against the disease itself, for example, ALS, to observe changes structurally or cellularly.
Yeah, the problem with these diseases is that there are no drugs. So that's why they are all incurable. There are some drugs that can ameliorate the symptoms, but there is nothing causal. On the other hand, I think what we are finding with our system can actually be helpful for the development of new therapies. So, for example, in this case, where we see that the proteasomes are impaired, that the other substrates are also not degraded, our collaborators have followed up on this, and they have shown that actually activating proteasome activity is helping ALS mouse models. So if we activate the proteasome in ALS mouse models, the mice do better. So this might be a new therapeutic strategy that's coming directly from our structural studies.
Great. Next question is from Anuruti. She's asking, does polyubiquitination play any role in the clearance of these aggregates?
Yeah, so the aggregates are in principle not clear. So we still do not understand, and this is something we are investigating at the moment. How do the proteasomes get there? It's probably through ubiquitination, but we don't really know what is it ubiquitinated, what's bringing the proteasomes there, because the aggregates are these poly-GA repeats. Ubiquitination normally happens on lysine residues, so there are no lysine residues. N-terminal ubiquitination is a possibility. So it could be that the aggregates themselves are ubiquitinated, and that's what brings the proteasomes there. But we know actually from other studies, from mass spec, for example, that many other proteins get stuck on these aggregates. And it might also be that some of these interacting proteins are ubiquitinated, and that's what brings the proteasomes there. So this is something we are trying to reconstitute in vitro at the moment. We are just trying to mix proteasomes and aggregates in vitro and see, what's needed for proteasomes to go there, and what's the structural basis of these functional changes in the proteasome activity.
Thank you. The next question is actually something that many of us might be wondering. What are the limitations of high resolution in cryo-ET, and how do you see the future development of cryo-ET?
What's the limit in resolution?
Yeah, this is asked by Yume.
Well, yeah, I don't think there is an intrinsic. So, I mean, you, Julio, know what Dimitri has been getting with his M software, right? So we already have structures better than, or not my group, but it has been shown by other groups that one can get structures better than 4 angstroms from entire cells by cryo-electron tomography. What's the limit for resolution is that, as in all cryo-EM, if you want high resolution, you need a lot of particles. When you purify them, it's easier to get a lot of particles, but in the cell, they are not that concentrated. So you need to record a lot of data if you need a lot of particles. So this is probably a bottleneck, but I don't think there is anything fundamentally limiting the resolution in this technique. And we are getting better and better technology and better software, and we are reaching the range of purified single particle cryo-EM. Another main limitation is what I mentioned before about tissue. We are now a little bit restricted to isolated cells. So we can look inside cells, but inside isolated cells. And this is something we are working very intensely to improve. And now I think we have a very good method to actually look into lots of tissues. This is something that we haven't published yet, but I think it's gonna drive the activities of my lab for quite a while. We found a very easy method in which we can zoom into a wide variety of tissues and address lots of questions related to neuronal function and neuronal dysfunction.
Mm-hmm. Thank you very much. So I think we will leave it there for the questions since we are a bit over time. Thank you very much again, Rubén, for the exciting and great talk. I would like to remind our participants that the next talk is by Claudia Futko. It's our third award student talk. So please join us there. And also, we would like to get your feedback on the conference. We are basically attaching this form always now in the sessions, and it's pinned. It's a handout, so please give us your opinion about the conference. We hope you are enjoying it. And thank you, Rubén, again, once for joining us. It's a pity we cannot have you here, but we hope to meet you in Göttingen. Thank you.
Next time. Thank you very much. Bye.